everyone, welcome to BTTC Science Corner Best Practice, and today we are going to make a DIY model of the lungs. Lungs are the most important part of our body, and we're going to look at how respiration occurs with negative pressure. For that we're going to take a sellotape, few straws, an empty plastic bottle, some balloons, a scissor and a cutter. Now let's look at how it is made. First we are going to make the trachea. I have taken one single straw and inserted two straws from below, and I have secured the opening with a tape. Next we are going to add a pair of lungs to this trachea and primary bronchioles, the right and the left lung. Two balloons are tightly secured with the help of a sellotape to the bottom of the straws. We are now going to take an empty plastic bottle which has a cap and it has the base. Cut the base, and this bottle will now act as the trunk of the body. We're going to pierce a hole in the cap of the bottle, and this is ready for use. We have opened the cap of the bottle, and the trachea, primary bronchioles and the two balloons which are the lungs have to be inserted from the base through the cap of the bottle. And we're going to close the lid securely and tightly, and using a sellotape we're going to seal off this opening. Please note that all the openings have to be tightly secured with the sellotape, and there should be no air entering. Now we only have to worry about the base of this bottle. So to make the diaphragm, we're going to attach a half-cut balloon at the base of this bottle. The balloon has to be slightly bigger in size and cover the entire base. Now our apparatus is completely ready for you. Due to the neural signal sent from the brain, the diaphragm is pushed downward. This causes a relaxation in the dome shape of the diaphragm, and as a result, the space between the lungs and the diaphragm increases. As the volume increases, the pressure inside decreases and air flows from out of the body inside the lungs, and the balloons get inflated. Now as the diaphragm contracts and acquires its dome shape again, the space between the lungs and the diaphragm decreases, volume decreases. As a result, the pressure increases and the air is released from the lungs and it travels outside the body. And this is how the interplay of the lungs, the intercostal muscles and the neural signals sent from the brain actually govern the process of breathing. Welcome to experiment number 2, and we're going to look at what is food adulteration. Adulteration is the act of making food of poor quality by adding some other substances to them. First, we're going to look at the adulteration of chili powder. It is the most commonly used spice in the Indian kitchen. Here I have taken non-adulterated chili powder, and this chili powder is adulterated with a common adulterant, which is the artificial color. We're going to do a very simple thing. We've taken 2 glasses of water with equal amounts of water in them. The glasses are transparent, and now what we're going to do is we're going to add the first sample into the water and see what happens. The chili powder soaks to the bottom and some of it floats. Now we're going to add the other sample to the other glass of water and observe what happens. Here we can see the visible difference in the color of the 2 samples. The adulterated sample shows a yellow color and the addition of an artificial color to the sample. The adulterants added could be as harmful as lead oxides, iron or copper sulfates. So next time we use chili powder, let's be more vigilant and careful. We are now going to look at the adulteration of salt. Salt is a major staple in the kitchen. It is the most important mineral, and we require it for conduction of nerve impulses, contraction and relaxation of muscles and homeostasis. The most commonly added adulterant to salt is the talcum powder to increase its quantity. Here I have taken 2 samples of salt which look almost alike. The left one is the non-adulterated and the right one is the adulterated sample. Again, we take 2 glasses of water with equal amounts of water in it. Both the glasses are transparent. We do a simple thing of adding the first sample to the first glass of water and we check what happens. We wait for a few seconds and take a spoon and stir the water. The water shows slight turbidity but is quite clear. Now we are going to add the other sample of salt to the other glass of water and instantly we can see that the water becomes more turbid and there are bubbles of talc which float on the water. This clearly shows that talcum powder is the adulterant that is added to the salt, and we can see a visible difference in the color of the 2 samples. We can see that the talc floats on top. The next item that we are going to look at is milk. India is the world's largest producer of milk, and ironically, milk is the most adulterated item in India. Most common adulterants are water, chalk, urea, caustic soda. I'm taking 2 samples of milk. This is the non-adulterated milk sample, and this milk sample has been adulterated with water. We're going to take a spoon and a steel plate which has a plain surface. We're going to take a few drops of the non-adulterated milk in the spoon and make it flow on the plate. We're going to check how the milk flows downwards. Now we're going to take a few drops of the adulterated sample and make the sample flow on the plate right beside the previous sample. As we can clearly see, the non-adulterated milk is viscous. As per the Food and Safety Standards Act 2006, selling adulterated food can be liable to penalty up to ₹1 lakh. If distribution of adulterated food causes death, punishment can be as severe as life imprisonment. Welcome to the third experiment, which is the biomimetics or biomimicry. It is the emulation of the models, systems and elements of nature for the purpose of solving human problems. As part of the biomimicry experiment, we are going to look at the nanostructure of the Colocasia esculenta, which are commonly known as taro leaves in India. If somehow we could mimic the structure of the taro leaves, which is like a honeycomb pattern, we can recreate a hydrophobic water repellent surface which will be useful in making window panes, car shields, etc. Now we have taken 2 leaves, one which is the Colocasia leaf and the other leaf that we have taken is the Ficus bengalensis leaf, commonly known as the banyan tree leaf. We have taken a bowl of water with a dropper in it. Now we are going to place 1 drop of water on the Ficus leaf and another drop of water on the Colocasia leaf. And we are going to simply observe the true drops of water and how they place themselves on the 2 leaves. Both the water droplets on both the leaves are more or less round. Now to test it further, we are going to take the tip of the dropper and spread the droplet on the leaf. On the Ficus leaf, as you can see, the surface is hydrophilic. It attracts the water droplet and the droplet becomes flat. Whereas when we spread the droplet of water on the Colocasia leaf with the tip of the droplet still remains round. This clearly tells us that the surface of the Colocasia leaf is hydrophobic and the water droplets do not stick to the surface of the leaf. This can be very useful in making water repellent surfaces. To find out why this actually happens on the colocasia leaf, using an electron scanning microscope, we look at the nanostructure of the taro leaves only to find honeycomb pattern which have elaborate ridges as seen in the right hand side image. The water droplets get gently placed on the ridges. The blue is the water droplet and the grey portion are the ridges. This increases the contact angle of the droplet with the surface, thus creating an obtuse contact angle on hydrophobic surfaces. If we use the same mechanism and mimic the nanostructure of the taro leaves in making window panes, car shields, etc., we won't have to use any additional waterproofing agents and chemicals. Biomimetics help us create sustainable, cost-effective, long-term and foolproof alternatives. Thank you.